So next, I have the rare and unexpected opportunity to introduce my mentor, Eddy Rubin, who's going to talk here about dark matter and beyond. And I think if anyone's been following Eddy over the years, you'll know that the one consistent thing in his research is simply going where the interesting science is, wherever it may be. And I think this will be a clear example of that, a really fascinating story from the microbial world.
Where's that pointer on remote if you want here? That's fine, I'll just sit here. Okay, it's a pleasure to be here. I love the science that users bring to the JGI, and I'm also very excited by the science that comes from within the JGI. And so it's really fun to have the opportunity to talk about some new science that's come from the JGI. And this is based on a project that Tanya Woki led that was finished at the end of last year, and it was a springboard to this project that I'll talk about. And so I'll give a little background. And so prior to the DNA era, before we were all born, before most of us were born, life was simple. You know, eukaryotes and bacteria. There was a trunk, and there were two big branches that came off that trunk. That's pretty much what you could see through a microscope. And then, you know, we entered the DNA era. And one of the first things that we learned was that there was a code. And we called it the canonical genetic code because it seemed like all life on the planet shared the same vocabulary. A little later, a very talented geneticist, Carl Woese, looked at little bits of data from the environment, from funny places, and he realized that the tree did not have two major branches that came off, but rather there was a third branch. And there's some debate exactly where this third branch came off, but the reality is that there really are three major domains of life. And it was really a very different view of life. And so the question is, at this point, you know, is there variation out there? Are there other domains of life? Are there other codes? And so that is sort of what has inspired this activities at the JGI, and the goal has been to look very deeply at, you know, if you want to find new things, you go to new places and you use new tools. And the new place is the uncultured world, and clearly if you want to find new animals, you don't go to the zoo, you go to the wilds, and that's the uncultured world. And the tools that we've been hearing throughout the day, and we'll continue hearing from this meeting, is, you know, metagenomics and single-cell genomics. There's a window into this uncultured world. And so we've been using these to go after what Tanya coined as microbial dark matter. And it's a term that's used quite, you know, physicists have this term, but now microbiologists are taking over this term. And so the metaphor for this is we've spent all of our time looking under the lamppost, and suddenly we now have tools to look elsewhere. And so what is the terrain? And the terrain is, this is a phylogenetic tree of all the known phyla. And the branches come from 16S sequencing. And in black are branches where we do not have a genomic representative. And so though we know they exist based on 16S, we know nothing about their biology because we can't culture it, and we know nothing about their capabilities because we don't have genome sequences. And that has really been the target of this project, to go after all those black branches. In gray are the branches where we have phyla with sequenced genomes. Okay, so Tanya gathered a group of JGI collaborators from around collecting samples from around the planet, really focusing on places where there was evidence suggesting that there were, you know, phyla that hadn't been sequenced in those environments. And these samples were brought back to the JGI, and it entered the JGI single-cell sequencing pipeline. And this is a pipeline that Tanya developed and has really been maintained by Rex Malmstrom at the JGI. And so we've been able to scale single-cell sequencing. And this is a pipeline that we're expanding because it is a window, you know, a powerful window into, or telescope into what's out there. And so the way this project worked is using sorting, and those of you visit the JGI this evening will get to see some of this pipeline. We were able to isolate 10,000 single cells, each in an individual tube. We then carried out whole genome amplification, a little bit like Steve Quake talked about last night. And then what we did is we characterized each of the tubes for the 16S marker. And we were specifically looking for tubes that contained genomes that had a 16S marker that didn't have a genomic representative. So we're really focusing on going after genomes that didn't have genomic representatives. And those are the ones that we sequenced. And from this project, there were more than 200 genomes at various levels of completeness that were generated and then characterized. And so a summary of the results are that in red are the newly captured phyla. And so these are the genomes that we never had represented previously, and black are the ones that are still out there. And there's wanted posters for them. And Tanya's very avidly looking for those genomes so we can complete this. But it's really an enormous phylogenetic diversity of organisms that we had no information about previously. And so there are a lot of insights that came from these studies and a whole series of discoveries. And I've been involved in lots of genome papers, and clearly this one had the most, usually genome papers are catalogs of additional genes. And this one had a bunch of insights that broke down rules, the transfer of genes between, archaea and eukaryotes never thought to occur, or eukaryotes to archaea never thought to occur. We showed that it occurred. So there are a bunch of really very exciting insights that came from this unexplored terrain. And we got the word dark matter in the title. And so now physicists, when they search dark matter, get to learn a little microbiology. And so there's a whole series of insights for this. But the one that really got my attention were the fact that we had, of these 200 genomes, 2 of them had remarkably small genes. The average length of a bacterial gene is around 900 base pairs. This one, these 2 organisms, these 2 Gracilla bacteria, had gene lengths of 200 base pairs. And we spent a lot of time scratching our head about this until Natalia Ivanova, who is a really senior savant annotator of microbial genomes, said, let's try other codon tables. And maybe that's why we're seeing such short genes. And so she used a table where the UGA, which is normally a stop codon, was now coding for an amino acid, was able to show that that's it. That you got normal-sized genes, that instead of being 200 base pair, you suddenly got genes of 900 base pairs. And so what's going on here is that when you annotate sequence with the canonical genetic code, and you assume that every time a UGA appears as a stop codon, you get 200 base pair genes. When you annotate the sequence using a different codon table, where now UGA is coding for an amino acid, you remove these stop codons, and you get 800 base pair genes. And then there are a variety of different criteria to, but you look at the translation product that really say, yes, this is really the correct code, and we were able to show that it was really a coding for a glycine, and it wasn't an artificial expansion. And so let me remind people of the canonical code. And there are 64 codons, 4 nucleotides to the third gives you 64. And of those 64 codons, 61 of them are sense, they code for amino acids, and 3 are for stop codons. And so that's illustrated here, and each of the sense have a tRNA that recognizes them and inserts an amino acid in the growing polypeptide chain. The 3 stop codons, on the other hand, have a releasing factor. So there are 2 releasing factors, and these releasing factors are structurally a bit like the tRNAs, but instead they terminate translation, and it ends the peptide. And so, but what's going on in this Opal recoded organism is it's chosen to decide that its UGA is not a stop codon, but instead is coding for a glycine. And so remind you that there was normally a releasing factor that recognized this UGA, and you don't want the releasing factor to terminate translation when it sees the UGA, and so the organisms got rid of that releasing factor. Instead, it's replaced it with a tRNA that recognizes the UGA antisense codon. And so it inserts an amino acid there. And so That's what's going on in this organism. And so this is the background for the study that's really novel that I'll be presenting, and that is that the microbial dark matter project, phase 1, expand the genetic representation of the tree of life on the path to providing an unbiased view of microbial life on the planet. And one of the other issues it brought up was it brings up the issue of the universality of the canonical genetic code. And what we've known about the code is largely from organisms that we can grow in the laboratory and animals that we can grow in zoos. But you know, how much is it the rule that's out there? And so obviously metagenomics provides a window into, you know, a massive scale window into uncultured organisms. So that's where we went. And so this is a project called Genetic Codes Operating in the Wild, and a whole series of informaticists from the JGI were involved in this. And though there was no experiments done in this study, there's a lot of biology that came from it. And so it's the ability to analyze sequence data. And so Nikos Kripides, Natalia Ivanova, Jim Tripp, Tanya Woke, and Patrick Schwentek was involved in this study. And this analysis metagenomic and phage data for codon reassignments, looking for codon reassignments in this metagenomic data set. And the data came from the JGI. In IMG, we have all the metagenomic samples that have been produced, the data from them. And so it's both a lot of human microbiome, various environments. There were more than 1,100 samples and more than 6 terabases of metagenomic and phage data. So it's really a very large data set that we went through. And what we were looking, there's a lot of places where recoding can occur. We chose in this study to look for stop codon recoding. And the reason for that is the most severe event. We have ways to really believe the data. There are ways to assess the data from various angles. And so we looked for recoding of the 3 stop codons in nature. And the way we did this was the way it was done in Tanya's study, in that we looked for gene size predictions and used 4 different codon tables. One table was the canonical code. Another table was where we said the Opal is now coding for an amino acid. Another table is where ochre, another table where amber. So we put the sequence through these 4 different coding tables and looked for evidence for that a different codon table is being used. And so basically when you use the canonical code, you're assuming that all 3 stop codons are working. You make your predictions based on that. The Opal codon table took away Opal stop codons, and similarly the ochre codon table, and likewise with the amber. And so a bigger view, we took 6 terabases of data. We assembled it because we want to see multiple events on the same piece of DNA, and we demanded that the DNA be at least a KB large, which led to 450 gigabases of data that fit that criteria. And then we put it through the 4 different codon tables, and then we had various filtering, quality control to provide strong evidence that when we were calling something recoding, that it really was recoding. And so what we found was that there was a lot of recoding going on. And this is in some way summarized in this slide. And what you're seeing is the color of the circles represent whether it's amber, orange, yellow, ochre, blue, opal. The size of the circle, the biggest size is really a greater than 1% of the sequence data have recoding. We see some samples, some environments where more than 10% of the data has recoding of stop codons. So it's really quite common, and where the circles are is where the samples were collected. And so we see a lot of recoding in humans and various places in humans as well as in the environment. Okay, next we looked to see where the recoding was from. And we looked for whether the sequences were from bacteria, archaea, or phage, where we saw recoding. And what we saw is that though we had lots of archaea sequence in our data, we saw no recoding in archaea. And this is not a small data set, so I think we may have set the boundary that recoding doesn't occur for stop codons in archaea. For bacteria, we saw exclusively opal. And that was really the biggest part of recoding we saw was opal in bacteria. And phage, there was one surprising finding, was that when we looked at phage, we saw opal recoding, which made sense, but we also saw amber recoding, which really had us scratching our head because we were wondering what these phage, what their hosts were. And so it was a puzzle. because there was a dogma that the phage need to share the same code as the host, that phage couldn't have a different code. And so we saw a lot of amber-recoded bacteriophage. We saw no amber-recoded bacteria. And so where were these phage going? And I'll give you the punchline. The punchline is that the dogma is wrong, that phage can, from these studies, phage don't really care about the codon usage of the host. They have ways of getting around that. In fact, they use differences to attack the host. And so I'll take you through a little diabolical story about this tongue phage. And this is a phage that we found in human metagenomic data. It was only found on the dorsal aspect of the tongue in multiple human samples. And in fact, we did our first clinical study at the JGI. We got IRA approval to swab the tongues of people at the JGI, and I think about 5 to 10% of them had this phage on the back of their tongues. And the phage is a 170 kb phage. It has it's a version of Veridia phage. It induces, and so this is a lytic phage, and so it produces its early genes, which sabotage the host's machinery, and its late genes, it takes over this machinery and produces its structural proteins, and it lyses the host. And what the clues that this phage gave us that it was operating this way was it encoded a releasing factor, RF2, which recognizes Opal stop codons. And this was the clue that told us how it was operating. And that, let me remind you about RF2 and Opal. When you have an Opal-recoded bacteria that I described earlier, it's now using its Opals as coding for an amino acid. And so it doesn't want releasing factor 2, which normally would insert a stop codon. And it doesn't want the stop codon competing with the tRNA. And so what the phage is doing, it's coming in, it's producing RF2. RF2 is now converting these UGA stop codons, UGA, which previously was coding for an amino acid, now to stop codons, and therefore was resulting in truncated peptides, 200 base pair peptides rather than 900 base pair amino acid proteins. And so when we look at the distribution of genes in the early and late phase of phage infection, the early genes is where RF2 is produced in the phage. So the phage gets in there, produces RF2. The RF2 sabotages translation of the Opal-recoded bacteria and host genes. In the phage late genes, it produces a tRNA that's able to recognize amber stop codon. We don't have an explanation for why this phage is amber recoded, but that is the observation. The late genes are amber recoded, and the late genes produce a tRNA that's able to recognize amber and insert an amino acid there. And so what's going on is you have this recoded, Opal-recoded host. It does not have RF2. And then you have this recoded phage, the tongue phage, that produces which has as a gene product RF2, early phage infection. The host is going about its business, translating the Opal as an amino acid, producing its products. The phage comes in, begins producing RF2. RF2 begins competing for the Opal stop codons, truncates the host products, shuts down the host machinery. Eventually in the late phase, the host machinery is no longer operating on the host. The host no longer has RF1. The phage now turns on its tRNAs that recognize amber, produces its proteins, and releases the virus. Okay, other evidence supporting. So we have no biology here, and so we're just sort of looking at the sequence data and trying to understand the biology from it. But other evidence supporting the amber phage infects bacteria with different codes comes from looking at the CRISPR-Cas system. And a lot of people, the non-microbiologists here, probably view this as Cas9, but it's an adaptive immune system that bacteria have developed to deal with phage. And the way it works is when a phage infects, squirts its DNA into a host, there's acquisition proteins that are produced by the host, and a successful host is able to break down the phage sequences and insert it into the CRISPR locus. And what you're seeing here in color would be phage sequences that have been inserted into the CRISPR locus. And so you see that these are really markers of phage that have invaded hosts previously. And so what Natalia Ivanova did was that she went through the 6 terabases of metagenomic data and identified all the CRISPRs, and then characterized them for the presence of the phage sequences as well as the surrounding host sequences. And so it's sort of giving us a footprint of what the host had previously been in contact with. And what she found was that there were spacers in there that had amber from amber phage, and so therefore it's saying that this organism had been infected previously somewhere in its history by a phage, and it figured out how to defend itself by inserting the phage sequences into its spacers. And then she looked at the surrounding sequences to see the codon usage of the host, and was able to show that the hosts around this had both Opal recoded bacterial genes as well as genes with the canonical code. And so it's saying the amber phage is able to infect hosts with normal code as well as those with Opal code, and providing very strong evidence that there is not a barrier, that the phage doesn't need to have the same codon usage as the host. And so in summary, the dogma that phage must share the same genetic code as host bacteria is not true, and in fact, phage can exploit the differences. And also, we see there's a lot of recoding in the environment, that a significant fraction of bacteria out there have recoding. In this study, we were just looking at stop codons. We're going to follow this up with looking at sense codons, see how frequent that is. There is a little bit of relevance for this concerning some of the directions of the synthetic biology community. There's been a great deal of interest in building recoded organisms, that they would be, there'd be a firewall between these organisms and the environment. And recently there was a paper from George Church's lab and Farron Isaacs where they recoded an E. coli, and they showed that it was resistant to infection with lambda phage. And from this, they felt as though they were creating this firewall. Well, I think this data suggests that maybe resistance to viruses, that there are a lot of viruses out there, phages, that can take advantage of these codon differences, and this may not be a firewall there. Also, the hope was that the recoded organism would not have the ability to spread its DNA to environmental organisms because it would share a different code. Well, this suggests that there's a lot of environmental organisms with different codes, and this probably isn't a firewall. Okay, and so where next? Well, this is, Tanya's now working up microbial dark matter phase 2. And this project is to look at 30 sites, and the hope is that many of these sites are chosen because there are reasons to believe there are deep sea brines and sulfur springs, that there's a lot of funny life there. And we're interested in trying to scale our single-cell sequencing, so to be able to explore that life. And in the past, we were looking for places, organisms that had 16S that we didn't know about. I think in the future we'll begin looking at organisms that don't have 16S and don't have, the thought that maybe there may be organisms that don't share share different codes as well as maybe new domains. And so we spend a lot of time spending money looking for life on other planets, unsuccessfully. My prediction is that we have a better chance of finding other domains of life on this planet. And so let me stop here and acknowledge Tanya and Chris Renke, the postdoc that really drove the microbial dark matter project, Nikos Kripides, Natalia Ivanova, Chris Renke, and Patrick Schrink, the postdoc that really drove the recoding in the wild. And so I'll stop and answer any questions. Thanks.
I think we have time for a few questions if anyone has any. All right, right here, do we have a mic to bring to her? Got one coming around so everyone can hear.
Is there any reason to suspect there may be recoding in eukaryotic genomes?
Yeah, there is. There are examples of that, and this study wasn't really to look for that, and we saw examples, in I forget the organism. But anyway, there previously were, and we saw that. And so there were, those were the ochres were. And it's not, it's in organelles, not in the genome.
Steve, we got one back there.
Have you had a chance to see if any single cell can toggle between two states under different growth conditions, for example?
That can go between one codon usage to another. You know, it isn't a mystery about how recoding occurs. And we see as organisms, their GC content changes. It becomes more efficient for them to use GCs for coding for amino acids and not wasting that on stop codons. And so that's where they lose amber and ochre. So I don't think they are toggling around. I think they eventually reach a point where they've given up and they get rid of the releasing factor. So I don't think they go back. And it doesn't occur all over the place. You can see very clear in phylogenetic trees that it makes this decision at this point, and all the branches that come off that point are recoded. Well, it has neighbors next door that didn't take that path.
So these recoded organisms seem to dominate or be most prevalent in uncultured clades. Do you have any reason to suspect that the recoding also correlates specifically with unculturability?
Right, right. And so there are examples. Mycoplasma has previously is a cultured organism, and it has recoding. And so we've known it previously, but we just thought it was a rare occurrence. And what we show that it's not so rare and occurs lots of places. And so I don't think it's, and I think the real explanation is that we have a very superficial view from just looking at cultured organisms. So there's a much greater diversity of uncultured organisms. And so therefore we have a better chance of seeing it there. The sampling issue.
All right, one more.
Yeah. Have you ever come across a case where you have two microbes that have the same 16S sequence, but otherwise they have a divergent genome?
Yeah, no, well, you know, obviously 16S is a very crude fingerprint, so you see very divergent genomes in organisms with same 16S's, but we haven't seen recoding in one versus another with the same 16S.
All right, I think we need to move on to the next talk, but thanks so much for that, Eddie. Let's give another round of applause.